Today I'm going to talk about lovastatin. In the 1950s and 1960s, it became apparent that elevated concentrations of plasma cholesterol were a major risk factor for the development of coronary heart disease. This led to the search for drugs that could reduce plasma cholesterol. One possibility was to reduce cholesterol biosynthesis and the rate-limiting enzyme in the cholesterol biosynthetic pathway, 3-hydroxy-3-methylglutarol CoA, or HMG-CoA reductase. Cholesterol is an extremely important biological molecule that has roles in membrane structure as well as being a precursor for the synthesis of the steroid hormones, the bile acids, and vitamin D. Both dietary cholesterol and that synthesized de novo are transported through the circulation in lipoprotein particles. The same is true of cholesterol esters, in the form of which cholesterol is stored in cells. Due to its important role in membrane function, all cells express the enzymes of cholesterol biosynthesis. Cholesterol is essential for the functioning of all human organs, but it is nevertheless the cause of coronary heart disease. Over the course of a century of investigation, scientists have developed several lines of evidence that establish the causal connection between blood cholesterol, arteriosclerosis, and coronary heart disease. Building on that knowledge, scientists in the pharmaceutical industry have successfully developed a remarkably effective class of drugs known as the statins that lower cholesterol levels in blood and reduce the frequency of heart attacks. The beneficial effects of statins are the result of their capacity to reduce cholesterol biosynthesis, mainly in the liver, where they are selectively distributed, as well as to the modulation of lipid metabolism derived from their effect on inhibition upon HMG-CoA reductase. These are the 5 steps of cholesterol synthesis. Cholesterol is synthesized in a series of more than 25 separate enzymatic reactions. First, there are 3 successive condensations of acetyl-CoA units to form the 6-carbon compound HMG-CoA. This is reduced to mevalonate, and then the polymerization of 6 5-carbon isoprene units to form the 30-carbon linear squalene, the immediate precursor to sterols. Then there is the cyclization of squalene to form the steroid nucleus, which then cyclizes the lanosterol, a methylated sterol. This is then further metabolized to cholesterol. A number of early attempts to block the synthesis of cholesterol resulted in agents that inhibited late in the biosynthetic pathway between lanosterol and the cholesterol. But the major rate-limiting step in the pathway is at the level of the microsomal enzyme that catalyzes the conversion of HMG-CoA to mevalonic acid, that has been considered to be a prime target for pharmacologic intervention for several years. This is the cholesterol pathway. HMG-CoA reductase occurs early in the biosynthetic pathway and is among the first committed steps to the cholesterol formulation. Inhibition of this enzyme could lead to accumulation of HMG-CoA, a water-soluble intermediate that is then capable of being readily metabolized to simpler molecules. This inhibition of reductase would lead to accumulation of lipophilic intermediates with a formal sterile ring. The statins do more than just compete with the normal substrate in the enzyme's active site. They alter the conformation of the enzyme when they bind to its active site. This prevents HMG-CoA reductase from attaining a functional structure. The change in conformation at the active site makes these drugs very effective and specific. Binding of statins to HMG-CoA reductase is reversible, and their affinity for the enzyme is in the nanomolar range, as compared to the natural substrate, which has micromolar affinity. The inhibition of HMG-CoA reductase determines the reduction of intracellular cholesterol, inducing the activation of a protease which slices the steroregulatory element binding proteins, or SREBPs, from the endoplasmic reticulum. The SREBPs are translocated at the level of the nucleus, where they increase the gene expression for LDL receptor. The reduction of cholesterol in hepatocytes leads to the increase of hepatic LDL receptors that determines the reduction of circulating LDL and of its precursors, intermediate density, IDL, and very low density, VLDL, lipoproteins. All statins reduce LDL cholesterol nonlinearly, dose-dependent, and after administration of a single daily dose. Now, these are some examples of the statin structure. Because statins are similar in structure to HMG-CoA on a molecular level, they will fit into the enzyme's active site and compete with the native substrate HMG-CoA. This competition reduces the rate by which HMG-CoA reductase is able to produce mevalonate, the next molecule in the cascade that eventually produces cholesterol. Lovastatin was the first specific inhibitor of HMG-CoA reductase to receive approval for the treatment of hypercholesterolemia. Lovastatin is an inhibitor of HMG-CoA reductase, an enzyme that catalyzes the conversion of HMG-CoA to mevalonate. Mevalonate is a required building block for cholesterol biosynthesis, and lovastatin interferes with its production by acting as a reversible competitor inhibitor for HMG-CoA, which binds to the HMG-CoA reductase. Lovastatin is also a prodrug, an inactive lactone in its native form. The gamma-lactone closed-ring form in which it is administered is hydrolyzed in vivo to the beta-hydroxy acid open-ring form, which is the active form. When lovastatin became available for prescription use, physicians were able for the first time to easily obtain large reductions in plasma cholesterol. Lovastatin at its maximum recommended dose of 80 milligrams daily produced a mean reduction in LDL cholesterol of 40%, a far greater reduction than could be obtained with any of the treatments available at the time. Equally important, the drug produced very few adverse effects, with once or twice daily dosing was easy for patients to take as well. For these reasons, lovastatin was rapidly accepted by prescribers and patients. After the early safety concerns were resolved by numerous animal safety studies and an intensive clinical research program that continued well past its regulatory approval, including a phase 4 study in over 8,000 patients, the efficacy and tolerability of the drug paved the way for its success. It's that lovastatin, once a potential orphan drug, went on to revolutionize the treatment of hypercholesterolemia. The overall benefits observed with statins appear to be greater than what might be expected from changes in lipid levels alone, suggesting effects beyond cholesterol lowering. Indeed, recent studies indicate that some of the cholesterol-independent or pleiotropic effects of statins involved improving endothelial function, enhancing the stability of atherosclerotic plaques, decreasing oxidative stress and inflammation, and inhibiting the thrombogenic response. Furthermore, statins have beneficial extrahepatic effects on the immune system, CNS, and bone. Many of these pleiotropic effects are mediated by inhibition of isoprenoids, which serve as lipid attachments for intracellular signaling molecules. In particular, inhibition of small GTP binding proteins, Rho, RAS, and RAC, whose proper membrane localization of functions are dependent on isoprenalization, may play an important role in mediating the pleiotropic effects of statins. That's the conclusion of my presentation. I hope that you enjoyed it, and thank you for listening.